What is up, my friends? This is a review video. This is just going to talk about what we covered in the last study session before spring break. So if you didn't make it to that, this video might be helpful. It's just here if you want to check it out. But the three things I'm going to talk about is we're going to contrast the two main cell types in the heart, which is the pacemaker cells versus the cardiac myocyte cells. And then I'm going to draw the electrical conduction pathway and kind of work our way through it. And then I'll just wrap up by talking about what is a functional syncytium. And there's three ways to say functional syncytium, or three ways to say the same thing. It's functional syncytium, functional unit, and myocardium. But we'll get into that. So let's start with the first one, which we're just going to contrast the two cell types in the heart, which we said was the pacemaker and the cardiac myocytes. So pacemaker cells versus cardiac myocytes. Okay, so the first thing is that pacemaker cells are autorhythmic. And this is because their main job is to generate action potentials. So, one second, let me just write this out real quick. I'm putting APs for action potentials. So, if you're creating action potentials, that creates a wave of depolarization and then contraction can follow. And so by being responsible for generating action potentials, you're really creating the rhythm or setting the pace for your heart rate. So yeah, these pacemaker cells really set the pace for your heart rate by creating their own action potentials. And this job is super important because we don't need nerves to create action potentials. With skeletal muscle, we needed to be innervated by nerves for our action potentials, but not in the heart. In the heart, we can create them all on our own with these cells. We still use nerves to either speed up the heart rate or slow down the heart rate, but we don't need them. We just need these pacemaker cells. Then we can create them. They create them all on their own. They just spontaneously depolarize. So yeah, just know their main job is to generate action potentials. Cardiac myocytes are contractile, and their main job is to contract or generate force. I'm just putting contract. And if we go back to block 2, if you guys remember those sarcomeres, which went Z line to Z line, there were those structures that shrunk when you contracted because the thick filament went up and it grabbed the thin filament, and then you had that power stroke, and then the sarcomere kind of contracted. So that's just important because it helps us know that since these are our contractile cells, we're going to have a lot of sarcomeres. And sarcomeres were responsible for striations. So striations, those were just the kind of stripes that you see in striated muscle. It gives it that striped look. So because its main job is to contract, you're going to have a lot of sarcomeres. But over here, the pacemaker cells in the heart, they're not really there to contract. They're there to make action potentials. So there's no organized sarcomeres. We don't really care too much about contracting. Okay. So I'm also going to circle this main job over here to contract, because that's really the main thing to know, is just that this cell type in the heart is for contracting, pacemakers in the heart are for generating action potentials. And there's a little phrase I use, it's just electrical conduction precedes contraction. Precedes contraction. It might seem obvious at first, but it's actually pretty helpful once we get into putting like our ECG waves onto a PV loop, or yeah, it'll be more helpful later, but just know this electrical conduction is what we're talking about when we're saying we're generating action potentials, because those action potentials create a wave of depolarization. So electrical conduction over here, the pacemaker cells, are needed before we can have contraction. So contraction was our cardiac myocytes. So everything that's happening with our pacemaker cells is preceding the contraction over here. So I'm just gonna draw a heart now, and we're gonna kind of work our way through the electrical conduction pathway. I'm gonna draw it in green. So we're talking about electrical conduction, we're talking about our pacemaker cells. So that's why I'm putting it in green so we can kind of see there. Okay, so this is gonna be not the best drawing, but okay. So that's our right atrium and our left atrium, and then right ventricle, left ventricle. Okay, so everywhere that I draw in green is going to show the location of our pacemaker. So to start, we have the SA node, and it's going to be up here in the right atrium. This is going to create action potentials, and these action potentials are going to depolarize both atria before they make their way to what's known as the AV node, which is right here between the atria and the ventricles. So that's AV. And then we're gonna propagate the electrical conduction down what's called the bundle of His. So this is this area between the right and left ventricle. It's called the septum. It just divides the right and left halves of the heart. You can think of septum as like a septum piercing for your nose, dividing your left and right nostrils. So the bundle of His is just that area being where the electrical conduction is being propagated down the septum of the heart. So I'm putting bundle of HIS. So it's going down this septum. And then once we get to these ventricles, we have these fibers. They're called the Purkinje fibers. And they kind of go down and around the ventricles, starting at the apex, which is the pointy ends, and then going up around the ventricles. So these are the Purkinje fibers. Purkinje fibers. So we just had the action potential traveling down the bundle of His. Now we're going down and around the ventricles. Okay, so that's really the pathway. SA node, atrial depolarization, AV node, down the bundle of His, Purkinje fibers, and then ventricular depolarization. So we're going atrium down and around, and it just kind of keeps going like that. That's our electrical conduction pathway. And now I want to talk about primary versus secondary pacemakers, because we do have two types of these pacemaker cells. So primary pacemaker. Our primary pacemaker is going to be the SA node. And it's primary because there's a faster rate of action potential generation. So when we see primary or secondary, don't think main versus less important, or anything like that. Really think of rate. So the speed of action potentials being generated. So faster rate AP, action potential generation. And because it's the primary one and it has the faster rate, it is the one that is responsible for our heart rate. I'm just going to put creates heart rate. Okay. Secondary, so that was primary and it was our SA node. Our secondary pacemaker is going to be, we have two of them. So pacemaker, the first one is going to be the AV node, this node right here. And then our second one is going to be the Purkinje fibers. And secondary, they have a slower rate of action potential generation. So that's important because, so this SA node was creating the heart rate really. Our AV node and our Purkinje fibers, those secondary pacemakers, they still create action potentials. They just do it slower. And so if something happened to our SA node, our heart rate would probably be a bit slower because they're secondary and they just don't create action potentials as fast. So that's really what we mean by primary and secondary. So primary is the SA node. And then again, the pathway started here. And as soon as we had action potentials being generated, they spread super fast through gap junctions. And I'm going to go over that next. And then they make their way to the AV node, down the bundle of His, and then through the Purkinje fibers. And electrical conduction precedes contraction. So everywhere we have these waves of depolarization, it waves through the atria, and then it goes down here. All of that happens right before contraction. So that's kind of why we have that small atrial contraction, and then we have that larger ventricular contraction when we talked about filling the ventricle with blood. So I'm just going to use the right side again for an example. So we said in the PV loop video and the cardiac cycle video, we had ventricular filling of blood, and then as soon as we had a little bit of blood left, we just needed a little squeeze. That atrial kick is what it was called, to get those last few drops of blood into the ventricle. And then once we have all the blood in there, we can use our cardiac myocytes and contract to create enough pressure. And as soon as the pressure in this leading side was higher than the pressure in the following side, the valves opened and we could eject blood out. So all of that is to say we had a little contraction there, and then we had a larger contraction in the ventricle. Well, that followed this conducting pathway, this electrical pathway. First, we had the electricity in the atria, and then we had that small atrial kick, and then we had the wave of depolarization, the action potentials being propagated down into the ventricles, and that preceded the ventricular contraction that we needed to pressurize the blood so that it could be ejected. Okay, so that's the electrical conduction pathway. Now I want to talk about this whole idea of gap junctions, because what we're going to see is there's a bit of a wall here. There's a wall between the atria and the ventricles, and we can have gap junctions in the atria and we can have gap junctions in the ventricles to spread these waves of depolarization in these two areas. But this wall is in the way. We can't have any gap junctions from the atria to the ventricle or ventricle to the atria. And that's why we need this AV node. Oops, I'll put it, well, it's that green AV node. That's our doorway for the electrical conduction to be propagated down to the bundle of His. If we didn't have this AV node, we wouldn't be able to do that because this fibrous skeleton wall is in the place. Fibrous skeleton. It's just a barrier, and there's no gap junctions in it. So that's really what we mean when we say functional syncytium. I am going to put a little definition over here. So, okay, functional syncytium. And what's a little bit annoying is that it has, I think it has two other names. So I'm just going to put AKA functional unit. At least those are kind of similar. And then myocardium. So if you see any of these functional syncytium, functional unit, myocardium, any of those, they mean the same thing. And what we're looking at, so let's just draw two cardiac muscle cells. If this is one muscle cell and this is another cardiac muscle cell, it could be any variation. So these could both be cardiac myocytes, they could both be pacemaker cells, or they could be one or the other. It doesn't matter, it's just anywhere in the heart. What you have, this area between cells, is called the intercalated disc. So it's kind of just that disc there. All you need to know about the intercalated disc is it's the location where you're going to find gap junctions. So in this disc, you have a gap junction, and it's really, just think of it like a pore between the cells. So gap junction. And what gap junctions allow for is they allow the action potential to be propagated cell to cell super fast. You don't need a nerve or anything. It's just, as soon as you have the action potential that was created from this SA node, you can spread through the gap junctions that electrical signal through the atria. And then you need the AV node to get to the ventricles after you had the electrical conduction, and then the entire ventricles can have that contraction event. But yeah, so this fibrous skeleton separates the two functional syncytiums. So what you can say is these green arrows, so the atria are one functional syncytium, or one functional unit, and the ventricles are our second functional syncytium. And it just means that we can spread our action potentials through gap junctions. The exact definition is, I have it written down, it's the area of the heart that can contract from one stimulation event. So if we had the stimulation event in the atria being this SA node, this stimulation event allows both of the atria to contract at the same time because of the gap junctions. So I'm going to erase my little drawing of this gap junction example, and I'm just going to write the definition just because it might help seeing it written out. But a functional syncytium is just the area of the heart that contracts from one stimulation event. And we only have two functional syncytiums in the heart. We have the atria and then the ventricles. So yeah, it just comes down to gap junctions really, and then this fibrous skeleton is the wall blocking the functional syncytiums. The only reason we can get it down into the ventricle is that AV node. But the other role of the AV node is also to create its own action potentials. It's just a secondary pacemaker. It's a little bit slower at creating action potentials than the SA node. So that's everything we really went over in our last study session. Next, we did go over one other thing, but I'm going to make a different video on it. Oh, sorry. I just had something weird happen with my camera. Oh, excuse me. And I had to cough at the same time. Okay. So the next thing we're going to do is just go through 3 different action potentials. We kind of had the skeletal muscle action potential already that we talked about through all of block 2. I have a photo of it right here, but we're going to go through these action potentials next. So we have the pacemaker action potential, which is this one, and then the cardiac myocyte action potential, which is for our contractile cells. And then in block 2, just to see everything side by side, we had the skeletal muscle action potential. And all 3 of these are going to be different. So that will be next. Okay, thank you for watching.